Genable technologies, innovative gene therapies for autosomal dominant genetic diseases. Let's quickly review the basic principles of our gene therapy for RHO-ADRP. Chromosomes are biological structures comprised of long strands of DNA, which contain the instructions for creating proteins, the building blocks of biological organisms. Zooming in, we can see that DNA strands are linear sequences, arranged into a double helix structure. Genes are short, defined segments of the DNA strands. The purpose of many genes is to store information for encoding proteins. Genetic diseases occur when genes mutate. A healthy eye allows light to pass through the lens onto the retina. Inside the retina, the primary structures detecting light are rod and cone cells. Rods specialize in night vision, while cones provide day vision as well as depth and color. RHO-ADRP can damage both rods and cones, causing partial or complete blindness in the patient. Impaired night vision, or an inability to see clearly in the dark, is one of the most common early symptoms of the condition, as is a gradual deterioration in the field of vision, even during the day. Depth perception can be affected, and the patient will be in danger of tripping, causing injury. Blurring occurs at the sides, top, and bottom of a person's vision. Daytime peripheral vision continues to worsen until the central part of a person's eyesight is the only remaining functional part. This is called tunnel vision. Gradually, even this limited vision fades, resulting in complete day and night blindness. Children of people who carry the defective gene have a 50% chance of inheriting the condition. Approximately 1 in every 30,000 people are affected. Treatments are available which can alleviate some of the symptoms, but there is currently no cure. The vectors will be delivered to the retina using a simple subretinal injection, administered by an ophthalmic surgeon specially trained in this technique. Zooming into cellular level, we can see how the two vectors bind on the cell's surface. At the center of the cell is the nucleus, containing the defective gene, which is causing the cell to produce a faulty protein, ultimately resulting in disease. The two vectors transmit their genes into the cell nucleus. The first cuts out the faulty gene, the second replaces it with a healthy copy. The aim of the treatment is to preserve those rod and cone cells that are still functioning. These changes should at the very least lead to an improvement in how quickly the disease progresses and could result in an improvement in vision. Our therapy has now moved from research to development, and we plan to commence clinical studies with the treatment in the next few years. Genable technologies, innovative gene therapies for autosomal dominant genetic diseases.